Good evening, everyone. Welcome back to the workshop. So today we are going to move ahead and we are going to talk about that what are the steps that we take when we have analyzed our raw rates and we have processed the FASTQ files and we have seen that what parameters need the improvement, right? So today we are going to work on that. My screen is visible.
Yes.
Okay. So yesterday, we discussed about that what are the ways using which we can fetch the different raw files. We discussed about few commands, and then we discussed that how we can do the QC of any raw file or any FASTQ file that you have for the analysis. Today, we are going to talk about that if we have some of the parameters that are not up to the threshold that are required, so how we can analyze them. So yesterday we were working on an E. coli data set. Today I have taken a sample from a human data set. Again, it was the same way as I'll show you how I fetched it. Okay. So again, this is a data set that I'm working here because I wanted to show that how we work with the human data set here. So you can see this is a project where a comprehensive assessment of RNA-Seq accuracy, reproducibility, and this is a benchmark data set basically. And you can see there are a lot of samples here. So it's the same way, just copy it. You can see this is the first, I have taken only the first sample here. Just copy the link, paste it in your terminal with the wget command, and you will fetch the files. When you fetch the files, it is going to be something like this in your system, which is a zip file. And when you simply click on it, or either you apply the command of the extraction, your files will be extracted and they will not be in the zip form. You can see I have my FASTQ files here. So again, okay. So here you can see I have my files. So let me quickly run a FASTQ. And if you remember, the simple command is fastqc. And then we type the name of the file, which is srr. Okay, it's a tab. And then forward dot FASTQ. Okay, the analysis is started. So just we apply this command so that I can show you how the results are for the particular sample and what parameters are there that we can improve here. I just did it for one sample. If you want to do it for more than one sample, so like you do FASTQC, type your first sample, and then after that you can have your another sample also. So depending upon the size of the data set, it is going to take some time, like it's already 65% done. Okay, so when it will be done, so as discussed yesterday, you will get a HTML file, which you can simply click and open in your browser, and it will show you about the basic statistics and also the parameters that the FASTQC has analyzed. Okay, so this is done, and I can go to my folder and I can open my HTML file here. If I want to look at where my file is, this is the file. Now, if you look at the file here, you can see again the basic statistics are there. You can see there are few green ticks and red ticks are there. Also, there is an orange one that is warning. You can work with that. There are reads, these yellow blocks, they are the reads. They are mostly in the green region. And then this is a warning region. This is the red region where your read should not reach. Everything is perfect except the per base sequence content is, and the type content is not proper here. You can see they are not at the same level here. But if you look at this GC content, you can see the yesterday that we were discussing, it was skewed. It has a triple peaks were there, but your GC content graph should be a bell shaped. And that is why today it is in the green parameter here. Then in content distribution, duplication level here and overrepresented sequence. In this particular one, we do not have the overrepresented sequence, but yesterday we saw that there might be some overrepresented sequence because of the PCR amplification. And here we do not have the adapter content here. Now, all of the parameters are perfect, just this sequence content and the tile sequence quality, which are because of the different levels of the base pairs that we have. So we have to improve this, right? Now, in order to improve this, we are going to move towards the trimomatic step, which will help us to improve the quality because this particular graph that we are looking at, which is of base sequence content, this may be because of some of the biasness or some of the contamination that is present in the base pair. So when we perform the trimming process, there are chances that these kind of one which are in red zone, they either move to the this orange region, which is a warning zone, but it is acceptable. And sometimes it moves towards the green zones also. Yes, Kanchi.
Ma'am, I just had a doubt. Like, uh, hello.
Yes, please. I am listening
Yes.
to your question.
Hi. Yeah. So I opened the Ubuntu terminal and wrote the same command as you, like fast QC SRR, but it is showing that the command fast QC is not found. How should I tackle that?
Did you install the app FastQC?
No, I did not.
Yes, that is the issue. Just simply on your browser type FastQC app on terminal. It will give you a command.
Okay,
So
okay.
first, these are like, if you work with Python or R, you have certain packages that needs installation beforehand.
Okay.
And also Trimomatic and HiSat2 feature count. These are all the packages. Like yesterday also, Jibisha at the end of the workshop, she shared that these tools need to be installed.
Yes, yes.
Yeah, so that is why you
Okay.
are getting that error. Once you have the FastQC installed in your system, you simply run it and it will be executed properly.
Okay, okay, thank you.
In the end, I can give you the command also. After we are done with this, I will share the simple command that you have.
Yes,
And in
yes.
simply, you can change the name of the tool and rest of the things will be same. Just run that and it will be installed.
Okay.
Okay. Now, after this, we need to perform some trimming here. And quickly, meanwhile, we are here, we can run the QC for the second sample also. One, and yes, we do not have it for the second one. So fast QC, SRR, tab, 2. Okay, so you need to analyze your forward and the reverse rate so that your trimming is applied to both the samples, forward and reverse. Because we are working with the paired end data, so it is important that all of the samples, like forward and reverse samples, they are both analyzed and they are trimmed here. As you can see in my folder, already trimmed file are present here, and also run the QC for the trimmed one. Because after trimming or after performing these pre-processes where we try to improve the quality of the data, again, we have to check the QC so that we can see that have our parameters improved or not. If they have improved, then we are good to go and we can move towards the next step here. Okay, a second or so, and then it will be. Okay, so this is also done. We can move here and we can see here HTML file. Here also you can see It's the same, just per base sequence content is not appropriate. Again, this is all peaked at the beginning here, at the beginning of the reads. You can see this is a position in the reads base pair. So at the beginning of the FASTQ file, the reads, they have this base sequence content that is not appropriate. So we can perform the trimming now. Now, okay. Now here again, what we are going to work with is, we are going to work with the tool which is Trimomatic. I'm going to share the command in the chat box with you. And then I am going to paste it in my terminal and I'll run it also and I'll show you here. Now I'll explain you what these parameters are. So now for this particular file, one sample that we have taken, there are two ways that we can go. If the quality of your file is very poor and you still need to work with it, there are some parameters, like we can go for some hard filtering. But here, what we are going to do is we are going to analyze with some of the linear parameters so that we have the appropriate results. Because sometimes if we go with hard filtering or hard parameters are there are some issues that most of the reads, they are removed or trimmed, and then what we get is just left over. So we need to decide that what kind of parameters we need to apply here. So here you can see this is a Trimomatic PE. Now Trimomatic is the tool that I was talking about. This is a tool that is used to trim so that we can improve the quality of the data. PE here, it stands for the paired end data, because we are using the paired end data, forward and reverse read. If you only want to work with one sample, like one read at a time, forward read only, then instead of PE, you can have SE, which represents single end. Now threads equal to 8, that means how many thread of the system we want to work with. I have done it 8. I can change it to sorry. I can change it to 12 also, because my system has that capacity. And then Fred equal to 33. So this Fred is representing the Fred score. So it specifies that your reads are using the Fred score of plus 33. That is common in the Illumina data. So sometimes like we have old data, when there is very old data we are fetching. Then instead of 33, we have to check that either we have to go with the 33 one or we have to go with the 64 one. But the latest technology that we are using, it make use of the Fred 33 here. So this is one of the parameter, and then you can see we are defining that these are our input files. This is my forward read, this is my reverse read here. Then I'm giving, then the next command define that I want to have the output files. So these output files will contain the trimmed data after the contamination or after what needs to be removed has been done. And then I'm going to get everything in the zip file format. And then I have this Illumina clip, TrueSeq 3 PE, this version here. So now this step ensure that there is a removal of the adapter sequences using the FASTA file, and this TrueSeq here. TrueSeq 3 is a file that contains the Illumina adapter sequences. And if this particular command analyzes that commonly used adapter are present in your read files, so it will match with it and it will try to trim it, right? So these are certain attributes, 2, 30, and 10 here. 2 means that if there are maximum number of mismatches, 30 here talks about the palindromic clip threshold there, and 10 is that simple clip threshold or minimum score to clip. So this is by default that you have to give it, that will automatically detect all the adapter sequences and it will trim the files. Then we have this sliding window 420. It is going to perform the quality trimming based on the sliding window here. So we are setting the window of 4 bases. And if the average quality in the window, it drops below 15, the trimming will start there, right? So this is how we give some of the parameters. Now again, if you want to change the values, like 4 is the basic one, like instead of 15 you want to go with 20, you want to go with 30. So again, you have to check what kind of quality we are looking at. If we are increasing the values, that means we are applying hard filters there, so it might impact the read files at the end. So try to begin with the lenient one. If the accuracy is not there in your data, then you can increase these parameters and you can check that. Okay, let me run this. Okay, it says, okay, it is running. Now this particular command, it is going to take some time here because we are performing the trimming. Now, when the trimming is done, you can see if you look at my output file here, so I'm going to get the trimmed 1P, 2P, 1U, and 2U here. And if I look at my folder, you can see I have 1P, 2P, 1U, and 2U in my file. So this is how we are going to get these different files after trimming. And when you have the trimmed files, you can again rerun the QC. Let me open a trimmed QC file. Okay, this is second one. Okay, so this was the first sample. And this is also the trim sample of the first one. If you look at the parameters here, you can see the per tile sequence quality was in red, per base sequence content was in red. But after trimming it, per base sequence content now is in front of it, there is a green mark. That means it is under the required parameter. And the one here, which was in the red zone, now I have gone to the warning zone. That means we can work with this. Now, there is a warning zone in the sequence length distribution because we have performed the trimming. Now, length is affected. That is why we are having this. And there is duplication level. If we see, duplication levels are not improved here. That means we can work with the parameters that we have. We can either increase or decrease it, and accordingly we can see that most of the parameters, they are resolved and they are in the acceptable region. And with some of the changes, will this work or not? So this is how we set the threshold for different files that we have. This is clear, everyone, till Tremomatic? And then also how to run the QC for the trimmed file is just the same command, fast QC, and then you give the name of the file. So this will be your trimmed 1P fast Q, and then you will get the same output as a HTML file, which you can see, and then you can compare your results with. Okay, after trimming and after QC has been done, now the next thing, I'll let it run because it is going to take some time. So we can discuss about the next step. After we have improved the quality of the reads, now next step comes of the alignment. Now for the alignment, what we require is we require a reference genome. And in this analysis, we also require a GTF file. Now I'll show you. Okay, can you see my ENA browser?
Yes.
Now I wanted to show you, there is, before alignment, there is one step which is indexing. Now, what is indexing here? Now, if we talk about the process of alignment, that means we are going to align the reads to the reference genome, and then we are going to make a comparison, right? But now before that, there is a small step where we perform the indexing. That means we are going to index the entire genome that we have, and we are going to convert it into more searchable or the compressed data structures. And today we are going to make use of the tool which is HiSat2. So HiSat2 here, okay. So HiSat2, if you search on the browser, you can see this is the tool, HiSat2 here. And if you see, this is one which you use for the alignment purposes. Now, when you go there, I'll share the link to this. Now, what is the purpose of HiSat2 here? Why I'm showing you this thing? Because I'm discussing about the indexing. So indexing is like that we are creating an index of a textbook. Now, now we are using HiSat2. What this alignment tool does is, it is going to search for that indexed file and it will try to move towards the regions here, or we can say it is going to where the reads they belong to the genome in a very quick and very efficient manner. So there is going to be index for the tool. So instead of going through each base pair, it is going to go to the exact position where the indexing coordinates are present, right? But for the HiSat2, they have a website from where you can directly fetch the files for number of organisms here. When you click on this, when you open this, on the right hand side, you have option to download. Because index, if you perform indexing, it is going to take some time. I was trying it. But HiSat2, this has the opportunity of predefined files over here. So just click on download, and here you can see you have few organisms for which the indexed files are already present. For humans, it is present, mus musculus, ratus here. Simply click on Homo sapiens, and you can see this is one of the assembly, 38. Just simply click on this. Either you can manually download it. Otherwise, what you can do is simply copy the link and use the command wget to fetch all the files. When you fetch it in your system, I'll show you. Okay, this is done. My trimming is completed here, and I have my particular files. You can see I have 2p file, 2u file is present here, and 1p and up is present over here. So this is completed. Now again, I'll just show you fast QC of any of the trim file. I just show you, I'm just going to give you a command, trim file, and this way you can do it. Non-model organisms as references. That means the organisms for which references are not present. That is your question, right? Wget. And you see, I copied that HTTP command from there. And with this, you can simply have all the files in your system. Now, if you see, I'm not going to download this because I already have it in my system. If you look at this, this is a genome index file where these indexing files are present. So I'll change my directory and show you what it contains. Okay. So it is going to contain something like this, .ht2, .ht, genome 1, 2, 3. So these are different files, and make a grch38.sh file. So these are the files that you can directly fetch, and then you can have it in your system for HiSat2 to directly work with them. Okay. Now, after this, let me see the question. Yes. So if you do not have a reference for your organism, then instead of reference-based transcriptomic analysis, you have to move towards the de novo one. So that is again another aspect of transcriptomic analysis, where you generate your reference genome from the read files that you have. That means you are going to use the FASTQ files, and using that you will generate your reference, and then you can perform this kind of like analysis. So, but today we are not focusing on de novo because that is again one of the technology within itself. So here we are focusing on reference-based assemblies. Okay, so after we are done with this, Trimomatic is done. We have indexing file. Now we'll move towards the alignment. Now, okay, I'm going to share the command in the chat box. Okay, So if you look at this particular command, it says we are using HiSat2. HiSat2 is the tool that we are going to use for alignment. X dash X is a prefix of the genome index that we are going to use here. If you notice that my files are inside the genome index, so I'm defining my folder. If it is not inside the folder, you have to skip this process. You have to skip this part of the code here. So I have everything inside the genome index / these are all the genome indexed files. And then I am defining, this is my input. -1, it is representing my input are the trimmed 1p and trimmed 2p files. And I want the output to be the SAM file. So yesterday, when I was talking about different file formats, so I discussed about the SAM, which is a sequence alignment file here, which is going to contain all the information about the read alignment in a text format here. So first we are going to get the SAM file, and after that we are going to convert the SAM file into the BAM file, because we cannot work with the SAM file, that is a huge one. We are going to convert it into the BAM, which is a binary version, and then we are going to perform the next step here. Okay, so when I run this, it's just a simple command. You have to define what tool you are using, where your indexing files are present, what are your inputs, and what kind of output you want. If you want it in some other way, like I have named my align.pair.sam. And this is, if you want to change the name, you can have it according to your will, and then it will give you an output file in such a way. If I show you here, you can see this here. This is align.sam file. Now I cannot open this because I have run the command that is under the execution here. So you can see after this is completed, you will get an alignment file like this. So I have this particular SAM file here. After this is done, what are we going to do? We are going to convert it, because conversion is one of the steps, because here we are going to use the SAM tools now. So SAM tool will help us to convert it into the BAM version, and then it will help us to sort also. Sorting will be there, and we are going to create the indexing. So using SAM tool, this is again one of the tools that we are going to discuss. Let me open the another terminal. This is under. Okay. Now I just run this inside a different folder. You can see this is the same command that I used here. And when it will be completed, you are going to see something like this, right? And if you focus here, you can see there is a, let me zoom here so that I can show you what kind of output you can expect after the alignment. When the alignment is done, you can see what is the alignment rate here, 98.22%. That means your trimming was apt, it was correct. And because of the trimming, your quality of the alignment has been increased. Now, if you it is the normal value that we discussed, that the alignment rate should always be greater than 90. And here we are getting 98.22. That means we are good to go towards the next step. So it is going to take some time to be executed. They have not given the time here. So our alignment is done. And now we have the SAM file here. If I do ls, you can see I have this particular SAM file here, which is going to be my input for the next step, which is the SAM tool conversion. So let me share the command here with you. Okay, so this is a simple step where what you are going to do is you are going to define your tool, SAM tools. It is not SAM tool, it is S. SAM tools is there. First, you have to install this. After installation, the very first thing is SAM tool views. So SAM tool views help us to change Now what we are defining that our input is a line.sam file, and I want to convert it into the BAM file. So this is one step. After the conversion has been done, I want to sort my aligned BAM file according to the coordinates. And then after that, I want to index them. So again, indexing is one of the important step here, which will create a type of the index, we can say. And it is going to give us the .bai extended file so that we can quickly load the specific regions if you want to, if you want to visualize it on IGV. So this BAI files help us to improve the visualization purpose, and also it will help us to look at the efficient features, like if we want to look at some genomes and exomes. So this indexed file will help us to look at that. So sorting has to be done, the conversion has to be done using SAMTool views. Then you have to sort it, and then you have to index it. These are important steps. So I have executed this. So again, it is going to take some time. Okay. After this is done, you can see you are going to get different output here. You are going to get the sorted BAM file. Then you are going to get your align BAM, sorted BAM, BA file. This is the index file that I was talking about. So these two files are there. So from SAM, you get the BAM file, and then you get the BAI file over here. Now, okay, so this is done. SAM tools aligned. Okay. This is completed. We have the file. Okay, here also you can see I just run the command that was showing to you. Again, the alignment rate is similar here. And again, you can move towards the sorting process. After sorting, there comes the step of creating the count matrix, like that I showed you on Gene Expression Omnibus, which is GEO. So that platform directly gave us the count matrix, where it talks about the gene expression values of the different IDs, the probe IDs, or the different genes that are present here, and it gives us the expression value. So to run the particular command, we have the tool which is feature counts. I'm sharing the command in the chat box. Okay, I'm going to run it here because I have a SAM file, BAM file is there, and index file is here. So I'm going to run it here itself. Now this is what the command is. Now here you can see we are using the tool feature counts. And then after that, you can see this is -A. It is for the gene annotation version. Now here we are defining that we are using Homo sapiens, the GTF file. Now, what is this GTF file? This is a gene transfer format here. And this GTF file, it gives us the exact coordinates of all the exons, all the features that are present in your dataset. Now, from where to fetch it, I'll show you. There are two databases that you can use for this particular GTF file. Now, whenever you are doing the feature count, you have to have this particular file because this will be taken as an input by the feature count tool. The first database that you can use is NCBI Genome. This is a database from where you can fetch all the different files. Okay. Here you can see we have a data for eukaryotes, archaea, bacteria, viruses, but we are interested in Homo sapiens. So I'm just going to run a search for that, and I'll show you what different files you can fetch it from here. Now you have to choose your assembly. We are going to go with this first assembly here, which is, and you can see this is 38.p. This is a version that they have. And now you can see we have the option of downloading here. You can manually download it. Otherwise, you can have the FTP link. So FTP links are useful when you want to use your wget command, right? And here you can see you have different file formats here, FFA, GTF file. So this is the file that you need where the genomic coordinates are present for all the features that are present in your data set. Also, if we talk about this FNA file, this is also used sometime. So for the genomics purpose also, you might require this particular database. But for now, we only need the GTF file. Simply copy the link, go in your browser, wget, paste the link, and it will be downloaded. The other database that you can explore is Ensemble. Ensemble FTP. Simply search Ensemble GTF file. Okay, FTP download. We'll click on humans. I'll share the link of this too. And here you can see you have option to download the coordinates here. And if you look at the gene annotation, you can download the GTF or GFF file. Here you can see again, you have this zip file. Again, copy the link. Use the particular command, which is wget. Wget and paste the command. It is a FTP link and you can fetch it. So it depends upon you that what database you want to choose and what files you want to work with. So these are two data. Sorry, I just had a power cut. So I'm on my mobile network now. Okay, so I was showing you this particular file. Just let me connect to the server once and I'll share my screen. Meanwhile, we can have some questions if anyone wants to ask anything. Okay. So once you have your GTF file in your database, directory, here you can see I have already this GTF file, and this FA is the reference file .fa extension, which is also the FASTA file. You can get it from Ensemble and you can get it from the NCBI genome itself. Now, if you look at this particular command here, so we are using the feature count and we are defining the GTF file. You can change it according to what the output you have. You can see it is matching my file name. And then I'm saying that I want an output. I'm defining that I want an output in the form of the count.txt. I'm using thread 4. And then I want, okay, and then I want to use my sorted.bam file here as an example. So when I run this, I'm just getting the add in between just a minute. Okay, so this is a command I'm going to run. Simply enter, and you can see the input is the BAM file, which is a compressed binary alignment file. Sorted BAM, then count. I want output as this. This is my annotation GTF file. I have used thread 4 here. And you can see this is creating a count matrix for me. This is not going to take much time as it is a one sample only. So here you can see the running time was 18 seconds only. And okay, my screen is hanged. So the output of this will be, you will have a count.txt file. Okay, let me rejoin again. From the trimomatics step, we produced 2 files with prefix trimmed. We are using trimmed P for downstream analysis. Could you help? No, we did not use. Okay, I'll explain you what this means. These are 2 different versions here, 1P and 1U. U are the independent files from the respective one here. So these are 2 outputs that are being generated. All of these are the trimmed version itself, but I have kept it as a different outputs here so that I can run my different analysis on this. If you want to have only one version, you can work with that. Okay, just a minute. I'm sharing my screen. I'll show you the count matrix file. Okay, so I run my command from here, count here. Count txt, if you remember, my command was about the output should be count txt, right? So this is how my file will be count txt here. Now this is the file that I've got. You can see there are different metadata informations available in your count matrix. These are all your ensemble IDs. These are ENSG. If you look at, just ignore this for now. On the left-hand side, you have this ensemble IDs here. You have the coordinates, and then you have certain values which are talking about the expression values here. So this is what your count file will be. In symbol IDs, different values on which strand it is present, and then what is going to be the expression value it is. Right? So this is how a count matrix is generated, and you have different samples, you create multiple count matrices, and then you have a combined file that is submitted to GEO, Gene Expression Omnibus. Or if you're using your own data, you independently create all the count matrix file, and then you combine them into one so that you have a data set prepared for your downstream analysis. So the purpose of introducing you to these commands was to get you familiar with the different steps and different tools that we have. So we need FastQ, we need Tremomatic, we need SAM tools, and then we need feature counts. So these are the tools that we require when we are going to move towards the automation step. So tomorrow, we are going to use the same tools itself, but instead of running each command like we did it here, we are executing each command at a time. We are going to make a script entirely, .sh script. It's a bash script, and we are going to have all the commands in it all together, and we are just going to give one single command, and using that your entire analysis will be completed at one go. So it depends upon how big of a file you have. It is going to take that much time. So this was all about the tools that we are going to use tomorrow. So tomorrow we'll discuss about the next flow. First few minutes we'll talk about the basics, the installation, and what are the alternates to the automation processes, and then we'll move towards the downstream here. And also we are going to share the script with you that we have created for automation, and then you can compare it with what we did as independent analysis, and then you can have your own script. Like if you understand one sample, then you can make it automated for your different samples or entire analysis at a go. Okay, so this was all, and we can take the questions now.
Yes, hello.
Yes, Ravindra.
Yeah. After getting this output file, count matrix, what else we can do? The graphs and all things. And
From the count matrix, like we did it for just one sample. When you have it for the entire project that you are performing, you can have the differential gene expression analysis. You can have the statistical tests like ANOVA, t-test, chi test can be applied in order to understand the differential gene expression analysis here.
So that thing we are not doing today.
No, we are not discussing that because we are talking about the automation of pre-processing steps till count matrix. Tomorrow, if we have time, I can give you just an introduction of how you can do that if you have the count matrix. Otherwise, we have a transcriptomic project. The course on the platform, it has a detailed description of various steps you can perform when you have the differentially expressed genes or you have the count matrix. So let me take you to the portal here. Just a moment. I can show you. And there we have different types of collabs also. Collabs in the sense we have different codes there. You can use those codes and you can understand about the steps.
See, it's, why I'm asking is, after getting this count matrix, we always confuse about like the how much p-value we can use.
P-value, see, P-value is fixed. We cannot change it. It has to be less than 0.05 to be significant. That is a statistical parameter. What we can work with is we can work with log FC. If you have seen like in few papers, the log full change, some uses of one.
That is my query. What should we like acceptable? What is the acceptable values and how much we can like this significant genes, how we can get those significant genes, the expression levels. Some papers shows different
Yeah, I
expression
understood your
genes.
question.
Yeah,
Some
yeah.
values are different. Some uses one. The basic threshold that should be that you should have a log FC fold of 1, at least 1. And then according to your data, you can change it to 2 also, 3 also. But 2 is like if we increase the number of the like threshold for the log fold change, that means we are moving towards the hard filter. So you can check. You can initially begin with 1. If you have a significant number, like you have got 100, now you cannot work with 100, so try to increase it to 1.5. If it reduced to 20, then you can choose your threshold as 1.5. If instead of 20, you are getting 200 at 1.5, you can simply increase it to 2 there. And your differentially expressed genes will be filtered at a much, we can say the hard filtering process will be there. So this is the course I was talking about. Here we have discussed about the basic central dogma quantification introduction. And then this is entire workflow that we have. Then you can learn about the QC. Then we have principal component analysis for the count matrix that you can use. This is a statistical test I was talking about. Once you have the count matrix, you can have different test codes here, why it is important, and we have also the R codes available here. This is all the theory. We have discussed that how it helps in hypothesis generation and all that. When you scroll it, here we have few videos also, and then there are going to be the collabs also for each lesson. Then Galaxy, okay, this is again here. Let me open this one.
Your screen is not visible.
My screen is not visible. Just a moment. It is visible now?
Yeah. Now it's visible.
So this is the workflow of differential gene expression analysis. First, we have introduction of what this is, how the parameters are there, and then you can perform differential gene expression analysis using different algorithms. And then this is all basic. After that, you can go towards the code here. So in this course, you will learn all about the downstream analysis. I'm just looking at the codes here. Here you can see, like once you have the count matrix, you can use the DSEC2 package, enhanced volcano, how you're going to load your data, how you're going to define different columns, and these kind of graphs or plots. You can have volcano plot showing you what are the differentially expressed genes. These are the ensemble IDs. It's similar to what we had in our count matrix file that we generated just now. Then you can have about the pathway analysis codes also. So everything here in this code is in detail with what is the concept of each path, each package, and what is its output, and also the biological interpretation is also added here. So you can check this transcriptomic course. And just here, this is the NGS transcriptomic complete RNA-seq analysis and the interpretation. This is also added to your program page. You can access from there directly. So this will give you the entire introduction of what can be done with the count matrix that you are asking, and how you can decide your log fold change. And again, I said that P adjusted and P value cannot be changed. It has to be less than 0.05. But what you can play with is your log fold change value. So start with 1 and then move like steadily increase your values. So that is how you're going to get the numbers there.
Got it. Thank you.
Okay. We are going to run it on the this system itself as we run the today's command. So not on the
Are there any more questions? Please feel free to unmute yourself or type it in the chat box.
Yes, ma'am. Can you share the commands to download FastQC and Trimematic?
yes, share. I'll share it with the commands for today, and you can find it on the resource pages.
Okay.
Any other questions regarding accessing the resources? If there are no further questions, no problem. You can reach out to us at support@omicslogic.com if you have any queries regarding the session or accessing the resources. And hope you enjoyed the session today, and we'll meet tomorrow at the same time. Bye bye.